All right, hello everybody, and thank you so much for coming to our Knauss Fellows Brown Bag Seminar Series today. Today I'd like to introduce Sam Chu Chin. He is a fellow this year with the Recreational Fisheries Initiative. Sam is a doctoral candidate in biology at the City University of New York's Graduate Center. He's originally from California and began his life in marine science as an undergrad volunteer at the Southwest Fisheries Science Center in La Jolla, and later worked as an observer in gillnet and longline fisheries. So please join me in welcoming Sam today.
All right, so we're going to take a minute to let the projector turn on. Thank you for that introduction, Katie. I put this presentation together recklessly and quickly, so hopefully it all makes sense, and please don't hesitate to interrupt me with questions. This is all I've been thinking about for the last 3 years. Thank you. So a lot of this, the technical stuff is going to be obvious to me, but it may or may not be to you. So feel free to jump in. There's the title of my presentation, Environmental DNA as a Force Multiplier. I chose this because I view the role of eDNA as enhancing other methods and other efforts that agencies like NOAA pursue. And it is a technique or suite of techniques that is a hot topic right now, and you hear a lot of hype about. People are saying it's a silver bullet, it's magical, it's a panacea, it's going to solve everything. We're never going to have to go sample in any other way ever again. I want to dispel some of that. As you heard, I've worked with some other methods in my life. I do think eDNA is good and useful and has a lot of applications, but I want to narrow its focus and remove the sort of magical sheen that it has, because believe me, it still takes hard work. Okay, so let's start off with, what is eDNA? Does anybody know what it stands for? Does it stand for electrical DNA? Environmental DNA, although in my case, it also stands for eel DNA because I use it to study eels. All right, yeah, environmental DNA. So what does environmental mean? It means we're sourcing DNA not from an organism directly. You know, we're not taking a fin clip or a liver sample. We're getting it out of the environment. So this is a method for acquiring information from the biosphere without actually encountering animals directly. Now you can use environmental DNA non-animals, but I'm a fish guy, so I only care about animals. Oh God, and I killed it. Oh, I fixed it. All right. Some examples of environmental samples that we can use. We use soil, water. Some people have done air, which blows my mind. And I recently learned at a conference that some people use honey, which I think is brilliant. They're effectively getting the honeybees to go out and do all the sampling and then concentrate it for them. And they can determine all the plant communities that are nearby, all the flowering plant communities nearby, based on the DNA they find in honey. Really cool stuff being done. So why is this interesting? Why do we care about the DNA that's floating around in the water? Anybody know? Anybody have any suggestions? Want to do my presentation for me? Well, living things leave behind DNA everywhere they go. You and I and everybody in here, it's a little gross to think about. You're leaving behind traces of your biological existence everywhere you go. Now, you've heard about this if you've ever watched a detective show in the last 25 years, or maybe a trial that happened in the mid-90s. We know that we leave DNA behind, and we know that we can relate that to who was there, who or what was there. Forensics has led the charge on this, and we've really gotten a lot of windfall benefit. eDNA is essentially windfall from forensic science. Being able to amplify trace amounts of DNA has given us this new ability. So what can this tell us that's useful? Well, we'll get to that. What problems can it solve? This is what I'm structuring my talk around. I want to answer these questions for you. Hopefully these are the kinds of questions that brought you in here or online. Okay, so what are some other ways we monitor marine life? Everybody in here has, or a lot of people in here, I assume, have done some kind of marine survey in their past. Shout out some ways that we do this. Scuba, yeah, so I got Sea Hunt up there because Sea Hunt's awesome, even though it's well before my time. Undersea vehicles, yeah, great. So we have like these automated drones going underwater. You could also have superpowers that allow you to talk directly to fish, but that is beyond, I think, most NOAA personnel abilities at the moment. I also have a scene from The Abyss up there. I'm from Los Angeles, so I make a lot of movie references. Hopefully that's okay with you. But yeah, I think the first way that humans ever decided to start surveying the marine environment was by looking real hard at it. So probably the first major technological breakthrough for marine science was like goggles or that tube with the glass at the bottom. They're like, oh, we can see what's under the surface now. So that was probably the first major technological breakthrough for marine sampling. We've come a long way since then in terms of looking real hard. Now we can listen for things. We can drag nets through there. We can catch fish. Tim and I work for the RecFish initiative. We talk to a lot of people that are like, oh, we want to do a citizen science project. We're catching all these fish. We know all this stuff. How can we make that data useful to you? All kinds of ways about figuring out stuff about what's underwater. eDNA is a fairly new addition to that, but I want you to put it in that context. Okay, so a bit about me. Katie did my bio earlier, but a little about me. So I'm actually still currently a PhD candidate at the City University of New York's Graduate Center, which is an old, beautiful building across the street from the Empire State Building. If you ever are in the city, look at it from the outside. It's super boring on the inside. I am currently a Knauss Fellow with the RecFish Initiative. I don't do anything with eDNA right now. I mainly deal with issues that relate to recreational fishing, or non-commercial fishing, as we call it in some places, which is a bit of a contrast for me since I got my start in marine science doing shark tagging, satellite kind of studies as an undergrad, and followed that up with commercial fisheries work as an observer in gillnet and longline fisheries. I followed that up with more gillnet sampling of kelp forests, and finally resorted back to that earliest of technologies, looking really hard at the water, doing some marine mammal mitigation work for the US Navy. This is not just biographical and self-serving. This is a way of illustrating, again, the many ways that we sample the marine environment. I've had the pleasure of working with a bunch of them. I've seen the pros and cons of several of them. Most of them are really hard, including eDNA, but they all chip in different ways. They all fit together different parts of the puzzle. So hopefully I can help you understand how eDNA fits that puzzle together. Sorry, my notes are on a separate page over here. So hopefully I'm not missing anything too important, but I actually promised that I will miss something important. Okay, so title of my talk mentioned cutting through the hype. And I think the best way to do that is to actually understand how eDNA works. I'm sure some of you are already familiar with it and are hoping I'm going to say something new and revolutionary here. Probably won't, but we'll see. For those of you who are not molecular ecologists, I'm going to give you a brief walkthrough through how eDNA analysis actually works, step by step. I like to break it down into 4 major domains. The first is collection. You got to go out there and get some stuff. You got to filter whatever environmental sample you have. You have to extract the DNA from it, and then you have to ask the DNA what kind of DNA it is. I'm going to go through these step by step here. Oh, that doesn't control anything. This controls something. Okay, so jump to filtration. Skip collection. Okay, so collection. You got to go out there and get some environment. If you want to do environmental DNA, you have to get into the environment or send somebody else out to do it. I do both. Getting out and doing it myself is more fun. Having a whole bunch of other people do it is more efficient. In this case, you can use very high-tech equipment like a rope and a bucket to do that, and a Nalgene bottle. This is one of the big benefits of eDNA is that the field equipment is dirt cheap and you probably already have it. The field skills are minimal. If you can dunk a bucket in the water and put it into a bottle, you're an expert field operator. This is a slightly more advanced version because I'm actually doing 2 steps at the same time. I did use a bucket, but I'm actually filtering as I'm collecting. It's a little bit more advanced because I'm advanced. This is for a whale project that I was doing up in New York. Okay, next step is filtration. This is an important step because this it achieves a few things. It actually captures the DNA-bearing particles on a little filter disc. Oh, and something I forgot to mention. Water, just because you put it in a bottle, the DNA is going to continue to degrade. There's still living bacteria in there. It's still above a temperature at which those bacteria can operate, and they will try to destroy the DNA that's in there. They like eating stuff. Cells, after they're dead, also try to destroy themselves. as the lysozymes break apart and the enzymes go crazy on the inside of the cells. So filtering it is a way of making it more convenient to carry around. Carrying around a paper disc is a lot easier than carrying around a liter of water that you have to keep frozen. It also really helps preserve the contents of your sample. What are we capturing on there? Some of it is what people call free-floating DNA, which I personally think of as sort of a myth. I don't think it's a meaningful contributor, but that's my opinion. That's you're going to catch cells that are out there. You're going to catch dirt. You're going to catch anything that doesn't fit through a filter. This is really, if you can make coffee, you can do all the steps of eDNA that we've talked about so far. In my experience, actually, dirty water tends to bear a little more DNA because the dirt itself captures some particles of cells and things, and you can extract that DNA later. So I've actually had the most trouble in the cleanest water that I've used. It also concentrates the DNA to a level where we can hope to amplify it later. One of the challenges of eDNA is that we're looking for stuff that isn't present in very high abundances or densities. So the more water you can drive through a filter disc, the higher concentration of your substrate DNA you're going to have, the more success you're going to have in finding whatever it is you're looking for. I already mentioned the degradation thing. So the next step is to now get that DNA off of there. I said you captured dirt, particles. Sometimes I can actually see little sticks or strands of algae on my filters. You should see the stuff we pull out of the Bronx River or the Hudson River or the East River. I work in not clean places, and the filters, they sometimes jam up and don't even let water through anymore. So at extraction, we use a variety of chemical treatments to burst open those cells and get the DNA out and remove everything else. There are a lot of compounds in environmental samples that will inhibit PCR amplification of DNA. Luckily, some big fancy companies have devised chemical means of getting rid of those for us. So I didn't have to do too much thinking. At the end, you go from a liter sample like this. I use a liter, by the way. People use all kinds of different volumes. 1 liter samples tend to be the most common because the bottles are cheap and 1 liter just has nice rhetorical appeal. Oh, and the projector's cutting it off, but you reduce it from 1 liter to this tiny little 100, little 100 microliter tube at the end of buffered extract. And at that point, this is a very stable sample. You can store these frozen or refrigerated even for really long periods of time, which illustrates another benefit of eDNA sampling is you can stop at several points and come back to it later. So you might have time to do collections, but you don't have time to do all your lab work. If you can get to this point, you can put the project on the shelf for years. Now, I don't recommend doing that because people like to see their grant funding come back and like to see results get produced. But if you need to, as is often the case, you can put it away and come back to it with really no loss of information. And just to underscore how conveniently these things can be sampled, what samples are these from? I think this is I can't tell what samples these are anymore. Oh wait, it says up there. Yeah, these are my Bronx samples. So this is an entire project in a single box. This was 10 sites that I sampled 4 times each, and all of it fits in a box. And these tubes are really way oversized for what's in there. They're probably 90% empty. I could fit this entire project samples in my pocket if I needed to. Now, reaching for an analog in other sampling methods for this is a little tricky. Like, I used to do a lot of gillnet sampling, and I didn't walk away from there with, I didn't store anything in a freezer afterward, usually. I walked away with sheets of data. And those sheets of data don't take up any space either, right? So you'd say, okay, that's more convenient. That's even more stable than something you're putting on ice. But this isn't the data. Remember, this is the entire sample. So this would be the buckets and buckets of fish that I would have caught in the gillnet and trying to store those on ice forever. No, that's going to take up an entire cold storage room. You're never going to store every contents of a gillnet that you get. So this allows you to store an entire biological survey in your pocket if you want. Well, I don't recommend storing it at body temperature. Okay, this is where we start getting selective. Up until this point, we've captured total DNA contents or total biological contents. The DNA extracts contain bacteria, human DNA, fish DNA, plant DNA, everything. But now we have to make a choice about what we're going to ask of the samples. There are two major methods we use to make inquiries of eDNA samples. We start with our extracted DNA that we just learned about. If we want to do, I think the one that you probably hear most about is metabarcoding. This is the one that says, who's in here? We use PCR primers that are designed to amplify a broad taxonomic range of species. The ones I personally use tend to amplify everything with a spine. They were usually designed for fish, but because of phylogenetics, you get everything that's derived from fish. So it's a nice little proof of some cladistic theory. The other big one is quantitative PCR. Now there are some methods that people use that don't neatly fit into either of these categories, but these are the two big ones, and they're all at least somewhat related to one of these. Quantitative PCR is PCR that's quantitative. You can count how readily something amplified in a sample. What this ends up giving you in the end is how much of a specific sequence was in there. This is, fortunately or unfortunately, depending on the purpose of your research, a drawback. You can only do a single species at a time. So instead of saying who's in here, this really asks how much of this one thing is in there. Metabarcoding returns a list of sequences. So the end result is going to be this giant file with a whole bunch of letters and numbers. And then you wash that against a reference database that you've assembled, and it tells you roughly how many of each were sequenced. Quantitative PCR, conversely, just tells you how much of that specific one was there. And to give you an idea of the difference in practical differences, this one, well, I always send it away for sequencing. So at minimum, this takes 2 weeks to turn around. Maybe if I had my own MySeq or sequencer, but nobody has their own sequencer. Some people do, they're lucky. This one you can turn around the same day. Because I'm lazy, it usually takes me a week to go from sample to qPCR result, but they actually make mobile qPCR machines that you can take into the field with you now and can give you results in as little as 45 minutes, including processing. Metabarcoding generally has a lot more steps in the lab, a lot more things to worry about. It's more costly. So you wouldn't want to do this for a small number of samples, but if you want to run a bunch of samples and you want to gather information about a bunch of species, this is the way to go. If you just want to know more about a single species, hey, is this invasive species here or not? qPCR is a great way to go. For my eel project, I mainly use qPCR because I'm just looking at abundance of eels, trying to see if it correlates with eDNA concentration. Oh man, the font didn't quite work. The slide is called a primer on primers. For those of you who haven't been neck deep in molecular biology for the last few years, you may not remember how primers work, but this is really the selective step of eDNA analysis. This is the first one where we have to make a taxonomic decision about what we're looking for. So just real quick, you know the DNA replicates itself, and it does that using RNA primers and enzymes that exist naturally. And the process of PCR amplification, which is really the basis of all molecular biology, makes use of these. And we can design primers, and this is the art of molecular biology, because designing the primers for eDNA analysis is tricky. For example, I think I have a bullet point for this. qPCR primers, because we're not actually doing sequencing in qPCR, we're only doing PCR, you need to do a lot of testing to make sure that they will amplify your target species and absolutely nothing else. So we had to work a little to make sure that our qPCR primers for my American eel project would amplify American eels reliably, and every time there was eel DNA in the water, but not do European eels, which are very closely related. So you have to find a sequence of DNA that varies between the two, and yet has primer binding sites that are conserved enough for you to routinely amplify. Another challenge with eDNA is you usually have to go with a short sequence because you're dealing with DNA that's often begun to degrade. So you can't go for a long chain like you might in forensic assays or like a 23andMe that you might use. You're going to want to find a smaller chunk because it's more likely to be intact in the environment. Metabarcoding primers are a little different. The ideal metabarcoding primer would amplify every living thing with equal frequency and affinity. Of course, that's impossible and will probably always be impossible. But the closest we've come is designing these ones for large taxonomic groups. We've found primer binding sites that are fairly conserved for, as I previously mentioned, vertebrates, and we can amplify those all. Well, almost all. We can amplify most of them pretty well. Some of them have mutations at the binding sites that prevent the PCR from working. A problem we run into in the fishes is that the teleos all amplify pretty well, but sharks, being sufficiently diverged, don't always amplify quite as well. So what you end up needing to do is use multiple primer sets if you want to get a more thorough coverage taxonomically. And finally, this is what eDNA results look like. Metabarcoding on this side. These are some of my qPCR results on the right. Now, this doesn't look like anything, so we usually turn that into graphics like this one. This is the results of my Hudson River Valley project. Each color is a different species. It's impossible to read from here. Don't try. It's actually impossible to read even if you blow it up to the size of the screen. This is a challenge that we're facing when you have an ability to survey a whole bunch of species all at the same time. You have new challenges in how to display that information effectively and meaningfully. But I think you can see there are a lot of different colors, and this is 23 different locations that we sampled in 3 days, which is pretty good, over 100 miles of the Hudson River Valley, and a whole bunch of species. And all those data came back at the same time. So you can really work in parallel quite effectively with metabarcoding. That's what that looks like. On the right, you have some of my qPCR results, and I'm actually not focusing on my own work for this presentation, but I'm gonna shine on a little bit for now. All I'm showing in this graph is that we have eDNA concentration on the y-axis and electrofishing results on the x, and it turned out that they were neatly correlated. So eel abundance seems pretty tightly correlated with eDNA concentration, which was a useful result. Yes, Amanda? Electrofishing. Oh, great. Yeah. So electrofishing, I thought I was going to be doing, this is like a different E method of fishing. Yeah, electrofish. The question was, what is electrofishing? Electrofishing is where you go into the water and pump thousands of volts of electricity in there to get the fish to come out so you could see that they were in there. It sounds like the most cruel way of doing it. It is the most fun way of finding fish. Absolutely. Of all the ways that I've caught fish, is by far the most fun. It makes me feel like a Ghostbuster. You go in there, and electrofishing really is one of the most comprehensive ways to do a fish survey, because it doesn't matter what taxonomic group you're from, electricity is going to get you, and it usually doesn't kill them. Actually, it used to, but then they figured out settings that are enough to cause a temporary seizure. So that you can scoop it up in a net. But believe me, we've lost plenty of eels, because as soon as we let go of the trigger, they jump out and they swim away very rapidly. Fish are tough. That said, we do miss fish, and sometimes their fish are buried or somewhere that they're just not going to be reached by the electricity. There are places that electrofishing can't reach, and eDNA is a way of coming at it at a different angle and reaching some of those. Also, you know, with eels, we're kind of okay electrocuting them. Some fish, maybe you don't want to. You know, if it's a really endangered species, maybe it's going to be hard to land a permit that says it's okay to shock the hell out of these things. So another useful thing. Yeah. Oh wait, that doesn't do anything. This thing. Okay, so now I'm just going to enumerate some of the strengths. This is a boring slide because it's just words. So with metabarcoding, you have really wide taxonomic sensitivity, and it doesn't require much prior knowledge. You can go into a place you've never been before, and you have no idea about the fish communities there, and you can have pretty high confidence that you're going to be able to recover which fish at least have left DNA behind there. Contrast this with a dive survey or any visual type survey. You know, I did all my visual work on the West Coast in Southern California. So you can put anything bigger than an inch in front of me, and I could probably identify it in about 3 seconds from like muscle memory, my brain somehow. But you put me, I've been working on the East Coast now for a bunch of years. I still don't know what the fish look like here. I am useless as a field visual fish identifier on the East Coast, to say nothing of places outside of America. But with eDNA, I can operate anywhere with pretty much equal efficacy because I'm drawing on the power of people that have submitted taxonomic identifications to the genetic databases that I query my results against. eDNA also has the ability to integrate its detections over wider spatial and temporal scales. So what do I mean by that? I really worded this terribly, I think. If you go, I'm going to make a lot of gillnet references because that's what I worked with before. If you go soak a net in an area and a fish swims around it, you'll have no idea that that fish was ever there, right? Only the fish that are stuck in your net are going to turn up in your results. With DNA, the fish can go around your water. I've never caught a fish in any of my buckets. I've tried. But I've detected a lot of fish that I haven't seen. So this really reduces the false negative rate. That's the main effect of this, is something that swam near your study site, you're going to catch. With electrofishing, for instance, we net off these 100-meter areas of river, and then we shock them, and we do 3 sweeps of shocking them. If there were eels right above or right below that patch of river, we would have no idea. We have no way of acquiring data about things that are nearby. eDNA doesn't have that problem because the DNA distributes itself through the environment. I have an asterisk next to this because you're going to see it show up on my weakness slide too. Another way of saying this is does not depend on direct interaction, which is also a strength and weakness because I really like interacting with fish. You can do some relative abundance work. As I showed with my eel slide, a lot of these measures can be quite tightly correlated. And so, you know, I could compare my different sites and say, okay, based on these eDNA results, we expect there to be more of fish X at this location without having done work there before. However, the literature does suggest that this really depends on the biology and the local environmental variables quite a bit. So I wouldn't bet the farm on quantitative aspects yet. A lot of work being done here. This is an area that's still in development. A big strength of environmental DNA is that it can detect and identify larval and gametic forms of fishes and other organisms, which is really something that, when it's possible visually, is extremely difficult. You know, identifying larval fishes for a lot of species, maybe even most species, there's like one person who's been dedicated their entire life to doing only that. Meanwhile, I can come in here, you know, 3 years into my PhD and go, yeah, sure, send me a water sample. I'll tell you everything that was in there. Now, I can't tell you if it was larval or an adult form. I have no idea about what form that DNA came from. But if I'm in, let's say, an area that's suspected of being the central fish habitat or a nursery ground, and we know that the adults aren't there that often, but then we detect eDNA in that region, let's say a seagrass bed or something, that's useful. That's potentially quite informative. The cost per observation is also very low. Because we can run so much of the analysis in parallel in the lab, you know, the cost of running 1 DNA extraction versus 2, 2 is not twice as hard as doing 1. Sequencing 1 sample is going to be actually quite expensive, but sequencing 96 is going to be the same. So you might as well, or roughly the same. It really is an economy of scale that we can leverage to our advantage here with eDNA because so much of it can be automated and use common laboratory equipment. Contrast this with, again, gillnet sampling. Every time you go out is going to cost you a whole bunch. It's going to cost a lot of manpower, fuel, whether or not you catch anything. As I said, it's readily automated. A lot of this stuff is still done the painstaking way, especially a lot of the pipetting and stuff, but we already have machines that can do it. We just have to build on that economy of scale and generate the interest in paying for those machines to do it instead of grad students. One of the biggest advantages of environmental DNA methods is their minimal field footprint. This is great from a variety of perspectives. I have a slide on this I'll touch on. And what I consider the biggest advantage is the partitioning of field work from the taxonomic assessment. You don't have to send your world-beating experts into the field, which again is a strength and weakness as somebody who likes to be in the field. Not that I'm really much of an expert. Okay, so some advantages of that minimal field footprint. It allows us to access new habitats that we might not approach with other methods. I have a picture here of, this is my advisor in front of me in a kayak. We're weaving our way into the interior of a mangrove. Anybody who's worked in mangroves knows you're not going to drag a gillnet through there. You're certainly not going to scuba dive through there. If you could stick your head underwater, it's probably so murky you wouldn't see much of anything anyway. And yet, I think anybody who knows about mangroves knows that they're pretty universally described as really important fish habitat. You know, in Florida, they're all protected. Zach and I were just at a conference all about them. They are quite important. The associative studies have shown, you know, that they might boost the productivity or biodiversity in nearby seagrass and coral reef beds, but we really don't know what's going on inside of them. eDNA might be a way of looking inside. Also, it's not just minimal impact on the habitat you're studying, it's minimal impact on what you're taking in there. It physically doesn't take a lot of strength to carry eDNA equipment around. That's a single suitcase. That was my entire field loadout for my mangrove trip. It was a suitcase of a pump and some gloves and some filters, and then another suitcase full of empty bottles, which really felt silly to carry around. But I didn't want to buy Nalgene bottles in Florida. Because of this minimal field footprint, in addition to new kinds of habitats, it can also easily be piggybacked onto existing surveys. It's not going to take up a lot of space on a research vessel. It's not going to take, it usually wouldn't actually require a dedicated person. If you have time just to throw a bottle of water in there or drop a Van Dorn in, which many studies are already doing for water chemistry or even just for temperature, you've already done the heavy lift required to do an environmental DNA sample. Really the hardest part about environmental field work is keeping the stuff cold afterward. So really this allows us to access places that are not accessible by other places, might be hazardous to work in, are protected. You know, my kelp forest surveys could never operate in some regions because they were MPAs. But that doesn't mean we're not curious about what's going on in the MPA. eDNA sampling is a nice way of not hurting things and still getting some information. It's not going to get every bit of information, but sometimes a blurry picture is better than no picture at all. And this is what I thought about the other day. It also is a cheap way of dipping our toe into investigating regions that just haven't sparked interest in funding sources or agencies. Maybe you have an area you think is interesting, but you just can't convince other people. Well, an eDNA survey is a really cheap way of showing, oh, this place is a biodiversity hotspot, or it's a cheap way of saying, nah, it isn't. Let's continue skipping it. As I previously mentioned, I think one of the biggest strengths is derived from the fact that you can separate the two worlds of field collection and taxonomic assessment. Your field techs don't need to be heavily trained, which as somebody who was trained by our very own NOAA, as once upon a time as a field observer, I don't like saying this because I was proud of my training and my ability to recognize fish and mammals and birds, but it is cheaper to send people with less training, or it is cheaper to go into a place and train people up there, or to train, you know, the public to go do some of this sampling. If you can throw a bucket, you can do eDNA sampling. You can even look like this guy who's wearing like a crunched up, beat up straw cowboy hat. And it also leverages the ability, the ability of a single lab. A single lab now can become this high power taxonomic assessment lab without moving anywhere. So it turns a lab into like this spaceship that can go around and scan for life signs. And there's a bit of a tiff between, in my world, between like morphological biologists and molecular biologists. You know, which way is better for knowing about evolution or identifying species, and molecular stuff can't tell you everything. And you'd think that I'd fall strongly on the molecular side because my dissertation's based on a molecular, has the word molecular in it. But really, eDNA is founded on the expertise of the morphological people who submitted these sequences to the databases. So what it really does is amplify their effect. If you're a morphologist, now eDNA frees you from having to make a bunch of field trips and identify the same fish you always see to concentrating on the most challenging specimens, and then you can submit your expert opinion to a genetic database in the form of a vouchered specimen and say, this is this, now use my opinion, it's the best. And now yokel like me can go out and use that as many times as he wants and benefit from the expertise of others. And this is, I had force multiplier in the title of my talk. This is what I'm talking about as a force multiplier. The morphological expertise of one person can now be used by anybody. Another advantage is that you can decide what to ask an environmental DNA sample after you've collected it. Contrast this again with a gillnet. If you're looking for fish, you're going to only look at the fish. Maybe you weren't paying attention to the invertebrates that were there. Maybe your gear wasn't capable of even catching the invertebrates that were there. With an eDNA sample, you might have an interesting fish result and say, gee, I wonder why this site had more of whatever species there. Does it have anything to do with the plant communities that were there? You can go back to that DNA extract and run it through the pipeline again with primers designed for plants and get a readout of what plants were there. Maybe your plant profile will be interesting, or maybe it won't. You could ask it about the inverts or the bacteria. You can ask as many questions as you have primers, so long as you don't run out of substrate. And my lab did this. I'm a fish guy. My lab mate's a bacterial guy, so we would use the same samples for totally different questions.
Are you subsampling what you're analyzing? You said as long as you don't run out of substrate.
Yeah.
So
usually we have about 100 microliters of DNA extract, and you only take 2 microliters at a time. So really you can run quite a few, unless you mess up a whole bunch of times, which I have done. Okay, so this wouldn't be fair. I did promise to cut through some hype, and it wouldn't be fair to attempt to do that without highlighting some weaknesses of eDNA. The first one I want to clear up is that eDNA does not actually detect fish presence. We usually describe it as if it does, but you should never forget that it's actually measuring fish DNA presence or organism DNA presence. Is this a good proxy for actual presence? Generally, probably yes, but not always. And I think a lot of the skepticism for eDNA can be cleared up by remembering this fact and keeping this in mind. Some people will accuse eDNA of being useless because of things like, well, you don't know if that fish was in that lake because it might have been eaten by a bird in one lake and then pooped out in another lake. Sure, that's true. But if you keep in perspective, the DNA perspective, that's actually kind of interesting. That tells you something about habitat connectivity if you have an animal bringing it from one place to another. We also can't do morphometrics. You can't measure a fish using eDNA. You can't even tell whether it was a gamete or an adult. And we're probably not ever going to be able to. Similarly, you can't do age classes. There's a lot of important information that fisheries management depends on that we're not going to be able to approach with eDNA. So it is not a silver bullet panacea. And even though it is a molecular ecology method, really right now we're not very good at doing population genetics with it. There have been some studies that have, and they've had some success. But generally, as I previously mentioned, the fragments are going to be pretty small. So we're not going to be able to do too well with things like effective population size. And we're not going to measure a whole lot of population scale variability. And as I also mentioned previously, the quantitative aspects are highly variable at best. Sometimes they work, but it seems right now the best way to approach these is you're going to have to do a baseline study and see if it works for that system and that species, and you're not going to be able to extrapolate across multiple species or habitats usually. However, within a region or a sampling area, relative abundance might work pretty well. You also can't discern where the DNA came from, as I mentioned with that bird example. Sometimes that matters, sometimes that might actually be useful. And as mentioned in my strengths slide, the spatial and temporal resolution of this method is kind of big. So right now, the best I'm hearing, the smallest spatial resolution I've heard is 10 meters, which is actually quite small. But usually the range often cited is, you know, anywhere from 10 to 150 meters. So you're not going to be doing really fine scale work with this. But as I also mentioned, that can be an advantage. So depends on your research question, just something to keep in mind. Also, the field work is boring. As one of my advisors often points out, he hates taking pictures of me squatting over a lake, because that's all it looks like. You don't have hero shots. You know, I don't scuba dive anymore, even though that's why I got into marine biology. The biggest weakness right now, far and away, for environmental DNA understanding and applicability and utility is that we actually just haven't done enough of it. We have a limited body for comparison. We don't understand where DNA is. You know, people say, well, you don't know where that DNA came from. No, we don't, because we haven't been able to look for very long. So this is the biggest weakness. It's also the biggest area for opportunity. The more of eDNA we use, the more we're going to understand where it is, where it might have come from, and what each individual sample means. Okay, a little bit about false positives really quickly. There are a few different varieties of ways that you can get a false positive in an eDNA sample. You can have what we call exogenous DNA, and that's where DNA comes in from an animal that wasn't actually there. So like my bird example before, I have a personal example I will tell you about. You might have sequencing error that throws, sometimes I get like weird protists in my fish surveys. Luckily, these are pretty easy to have the computer filter out. Like if it's something that isn't anywhere close phylogenetically to what you're looking for, probably just some kind of sequencing or amplification error. Sometimes PCR can stick two sequences together, and then it will read like this half of one species and half of another. Again, these are pretty easy to filter out bioinformatically. Sometimes your qPCR, you know, if you're not careful, as I previously mentioned, you will amplify the species you're looking for and maybe some other species, and then that will throw off your readings. So that's an argument for really doing a lot of prior testing. False negatives are pretty low. However, PCR bias does come into play. As I mentioned with designing primers for both teleost fishes and sharks, you know, I did some aquarium sampling early on, and I was using these primers in tanks where I could see sharks, and my samples came up and said, there are no sharks here. And I knew that was a lie. But the approach that is generally used to combat this is just to use multiple metabarcoding primers simultaneously. You also might not see something that you know is or expected to be there because it hasn't been uploaded to a genetic database. For me, this has been hickory shad. I knew there were hickory shad in the Hudson River Valley, but for some reason in the year that I was doing that survey, nobody had ever uploaded a sequence for it. But again, this is something we can fix post hoc. We can go back, build a new database, ask the question of the data again, don't even have to do lab work again. You also might have a circumstance where the organism really isn't emitting DNA, but it's there. My eels, again, may be an example of this. They go into this hibernation phase in the winter where they shut their bodies down almost completely and bury themselves in the mud. They don't really release a lot of eDNA into the water at that time. So you could say this is a false negative because the eel is there, but you're not detecting it. However, again, think of the DNA perspective. It's kind of interesting that it's not emitting DNA. And if it's not emitting DNA, how biologically relevant is it in the water at that moment? Here's a little example from my Hudson work. So I only focused on these tributaries of the Hudson, not the Hudson itself. So they were all freshwater and they were all above tide line. And yet these 5 saltwater species showed up. I had hake, scup, mackerel, salmon, and swordfish in the Sawmill River. I know this is NOAA, we mainly work with saltwater, but so you might not know, the Sawmill River does not have a billfish population. But what it does have, so here's where I sampled it. And here's a place called Ocean Fish Market. So this highlights, again, what I think is both the strength and weakness of these methods. Alone, I would have no way, if I had never been to this area before and I didn't know anything about it, I would say, yeah, I guess there's swordfish in that sample. And then somebody would say, you're wrong, I'm terrible. But with a little bit of extra information, you can now see something that's kind of biologically interesting. So I originally thought of this as just a total failure. I'm like, oh man, this is terrible. You know, a gillnet never would have thought there was a swordfish there. This is an inferior method. But at its root, eDNA sampling is chemoreception, right? We're looking for, I often call it a genetic nose. We're smelling for sequences of different species. And then you think about it, fish olfaction is millions of times better than ours. I'd be willing to bet that the fish can tell that there are signatures coming from this fish market and getting into their water system. Now, I'm not saying they're smelling the sequences that I'm detecting with my assays, but there's probably some sort of sensory effect. So is this a perfect analog for a gillnet? No. This is subject to errors that gillnet wouldn't be, but it's also able to sense things that might be biologically relevant that no other method can detect. So I really want to use this to reframe how we think of eDNA. It's not a new way of running a gillnet or a new way of doing a dive survey. It's a fundamentally new way of sensing the biosphere. We are visually oriented animals, so we like to see things. That's the most convincing thing we have. However, a lot of the underwater world is not terribly visually oriented, especially the waters I work in. There's not a lot of light getting through. It's very dirty. Olfaction, hearing, those are often better. And this is really a way of approaching that exceeds our innate sensory abilities. I called it the genetic nose. And this is also still a technology that's really in its developmental infancy. So a lot of the noise in the data that is coming out, I don't view it as a bad thing. I view it as figuring out, you know, those are keys, those are clues to what the future might be. We don't know what puzzle we're building right now. We're still piecing it all together. I don't know if you guys watch or read this comic. It's on Instagram. It's called The Strange Planet. There's this great one, just this little baby alien kid going, "Chaos is how I learn." You know, a lot of the talk about eDNA, especially at high-level meetings, is how can we standardize? Let's standardize this so we can all do the same thing and get, you know, comparable results. I don't think we're ready for it, and I don't think it's a good idea right now. I think right now we need to mess around as much as possible to figure out what kind of information we can collect, what we can see, what works and what doesn't. I also want to push this line. Think of eDNA as a supplement rather than a substitute. Don't ask, can I use eDNA instead of? That's the wrong way to approach it. I think the right way to approach it is to say, can I use eDNA to improve upon or to augment my surveys? If I have a gillnet survey that includes an area that has an MPA in it, well, can I just use eDNA and maybe gain some information, fill in those gaps a little bit? That's a more effective strategy. Of course, we're going to have to always draw comparisons to other methods because this is what allows us to trust anything. But I think the real gains are going to be where we don't have overlap. So what does eDNA offer that's fundamentally new rather than what does it do that other methods can do? Another analogy, so this is the first satellite photo ever taken. It's terrible. This is a terrible photo. This gives us no information about anything, but it was taken in 1959. If we had said, oh, you know, that doesn't tell us, that's not going to help us, you know, find sailors lost at sea or define, you know, the extent of whatever, and we've given up on it, that would not have been a great strategy. This is one I pulled down 2 days ago. We've come a long way, and I would expect a similar arc of development for environmental DNA. Right now, we're really the picture on the left. We have a blurry, incomplete, you know, picture of the genetic biosphere. I hope to have a much more comprehensive view coming up. And yet, our pictures of today of the genetic biosphere are actually kind of cool. This is a study done 3 years ago, and they did a bunch of trawls, and they worked in tandem. They did eDNA and they did regular capture trawls, and they interpolated the data and developed these heat maps of fish presence. And I think this is a super appealing way of looking at environmental DNA data. But imagine if that was filled in a bit more. And now imagine if they had a time series and you could see range shifts throughout that time series. That could be really powerful. You could define or help define fishing seasons based on that, or fishing closures if you have a fish you need to protect. So what can we expect the future to bring for eDNA? I think both of those things I mentioned, more time series data, broader spatial sampling. Now, what can we do now to hasten that future, to make it more useful? I think the best way to do it is just to use more of it. It's easily piggybacked. It doesn't take a lot logistically to add it to a study. So please give it a try. The more puzzle pieces we assemble, the more we'll have an idea of how they all fit together. As anybody who's done a jigsaw puzzle knows, the last bit goes the fastest because you start seeing what colors should go where and what things mean. Each individual piece gains power based on assembly of the others. Of course, promising that it's all going to be great in the future is not useful. People want something to help them now. So current capabilities are really going to drive wider adoption, which is going to in turn drive more utility in the future. So the next few slides are just some ideas I kind of threw together of how they might fit into some NOAA type projects. This is a little bit of a flow chart. So is eDNA right for you? Should you think of using this in your own work? Are you happy with your survey methods? Do they tell you everything you want to know? And can you do them with the frequency and taxonomic coverage that you want? Then you don't need eDNA. You've got a perfect life. But you still have an opportunity to help me out by doing a calibration or validation study. You drop an eDNA bottle in there, run a survey. Hey, you can help me out. We can see if those numbers match up. If they don't, that's also interesting. If you want more from your surveys, if there's a gap in your knowledge, maybe we can fill in that gap using environmental DNA, depending on what your research question is. Okay, so here are some real quick ideas I had. You can use eDNA to target the use of other methods. You could use qPCR, do a qPCR dip, use that mobile on-site machine. Decide if you want to send your divers or drones or submersibles or whatever down, or if you're going to get skunked, or you're next. It costs a lot to field some of this equipment, and it sucks when you spend the money and you get no data. So this can allow us to do a little bit more with less or the same amount of money. We can also discover accidentally biodiversity hotspots through metabarcoding. The agnostic abilities of metabarcoding allow it to accidentally find things we weren't looking for. We can study habitat connectivity because of our ability to detect gametes and larvae of species. Also, the ability of DNA to persist in environments that sometimes, like the bird example, tells us something about how environments might be connected. This is also, I'm going to hit mangroves and seagrass and corals again, because this is a good opportunity to pair eDNA with other methods. The clear water around corals and seagrass is great for visual sampling. Get in there, put a mask on, look real hard. Then compare it to what your eDNA is in the mangroves and see if that's interesting. Also for habitat restoration, I've had the pleasure of working with a little bit of oyster, or funding some oyster restoration through my RecFish time here. I also worked on some in my graduate work. If you're restoring an area, you probably don't want to drag a gillnet through it. So running an eDNA sampling might be a good way of seeing the progress in the restoration of those communities. You could do large-scale surveys. I did over 100 miles of the Hudson River Valley in 3 days. I thought that was cool until we were at this mangrove conference, and a guy there revealed that they'd done the entire coastline of Japan in a year. Japan has a lot of coastline. This isn't published yet, so I don't have the citation for it, but that's what they said, which is really amazing. The Japanese groups are doing some amazing work with eDNA. And so this also gets at the strengths of it. I said that the spatial resolution isn't very good, so why not go big instead of trying to go small? It scales up very nicely in ways that gillnets and visual surveys don't. You compare acoustic surveys with eDNA. Acoustic surveys are really good at high-res biomass, not very good at determining and identifying which fish are actually in there. You put those together, you have the best of both worlds. This is also a Japanese study that paired the two together. More of a comparability study, but again, developing a heat map, nice work. You can monitor range shifts for not a lot of money across a pretty wide spatial area. This is work on the right that a guy on my dissertation committee put together. You can see the changes. These are all species of fish. These are months right here. And you can see how the seasonality changes for those populations. I have a picture here of a black sea bass that some of you may know they've been showing up in lobster traps, which they never did before. Could also do invasions. It's also an opportunity for citizen science. Like I said, anybody can throw a bucket in the water. This is something that people have begun to leverage public interest into actual scientific collection. And this is really a good way to engage with the public and engender more buy-in and interest in ecological kind of work. And you can also just get more samples that are useful. So this is a crayfish study they did in the Great Lakes. This is a newt study they did in the UK. And both used like dozens of volunteers who worked for free and still got pretty good information because the error rate for field operators for eDNA is pretty low. I also think this is a good way of maybe getting some better scientific information for really data poor regions or species. There are species out there that this agency is charged with monitoring, but we can't assess formally because we don't catch them enough or encounter them enough, or they live in places where we just don't have the resources. But if you can drive a boat through a region and collect water, you can at least get some presence-absence information. Is this going to give you a great structured stock assessment? No, but some information. A blurry picture is usually better than none. And so to summarize, this is a supplement, not a substitute. Think of how it can add to, not how it can replace. If you can't catch something, let's try to eDNA it. Let's try to use this to reach into new locations. The more of this we do, the more useful it will be to each of us. This is really an economy of scale. And I really want to promote time series and grid form sampling. That's going to be more convincing than any static sampling or point sampling would be. And this stuff is cheap, about $10 to $50 per sample. Your field equipment you probably already own. So if you're interested in this, give it a try. It really doesn't cost much. I will put you in touch with people or I will do it for you. Anybody have any questions in the 2 minutes we have left? Sorry, I went long.
Thank you, Sam. Any questions?
Yes.
You highlighted that DNA degrades pretty quickly, especially once it's been sampled. And I'm wondering how long DNA lasts in the open ocean.
Yeah, so the question is about how long DNA lasts in the open ocean. There are studies about this, and degradation doesn't seem to be the primary driver of loss of signal so much as just dilution. The ocean's so big that within 150 meters or so, it dilutes to an extent that you're not going to be able to amplify it anymore.
Within what?
About 150 meters is what I've read.
Okay, and what about temporal scale?
So that would assume, I mean, it's hard to separate them, right? Because it's going to be hard to find a piece of ocean that doesn't move. But people are looking into it. I can't give you a number right now. Any other questions?
Okay, so we did have a couple online questions.
Hello, Internet.
Someone wanted to know, to do qPCR, do you need to filter known volumes of water from each sampling site?
Yeah, it's definitely better to standardize your volumes of water for qPCR sampling. And for all eDNA that I know of, you do need to filter or somehow precipitate out your DNA.
Anyone else in the room?
Which manufacturer's filtration work out best for you?
I use Illumina and QIAGEN products. If they want to sponsor me, that's cool.
How do you go about reducing the scale?
We might not be able to. It may be that these things are just too mixed, right? However, there was a paper by the Wilcox group, I think, that shows, you can be a little bit selective by varying your sample volume and your pore size. So in some cases, you might expect that larger particles particles are going to break down, right? So the larger the particle, perhaps the more recently it was emitted. So if you only sample large particles in small volumes of water, you're going to be integrating over shorter timescales and smaller spatial areas. So that's one way of cranking it down a little bit.
And someone else online wanted to know, when you factor in the cost of lab work, including sequencing, is eDNA still cheap? In other words, are the costs of field work or more traditional methods that prohibitive?
Again, it's an economy of scale. So if you just want to sample one area, probably not. But if you want to do a whole bunch of areas, actually with qPCR, yes, it's cheap, no matter how much you do. With metabarcoding, you want to do a bunch before it really becomes cost effective. A guy, Mark Stokel, who I quoted in my paper, he's also developed his own assay called GoFish, which is a really, really cheap way of doing it. A great way to get into eDNA sampling and analysis without a lot of money or special equipment. And I think he got it down to like $10. And it doesn't need an economy of scale. You can do $10 your first time. So and he did that great paper by throwing a bucket off the railing at the like lunch pavilion at Rockefeller University.
All right, so we're just going to adjourn the webinar for now.
All right.
And then, so people online, if your questions didn't get addressed, I will forward those to Sam. And so thank you so much.
Thank you. Goodbye, Internet. All right, now what do we